on here as well, because I do want it to be available for people, because a lot of my students as well are doing chemistry along with this. So I'm assuming they're studying for that right now, right? Hopefully that's going to not glitch on me. You can go away, I don't care about you. Stop you. Right, okay, we're back, I hope. So if we are therefore looking at this question here about the structural gene and about the regulatory gene, what's the difference? Structural is going to code for enzymes, is going to code for these structural proteins, whereas regulatory genes, that's going to code for proteins that are required to either stimulate the expression or inhibit the expression. So what you want to do is you want to ensure that you understand that it will either actually form a protein which is a structural, an enzyme that's required, a antibody, for example, a actin or myosin if we were looking at the muscle. So it's a structural gene. It's going to produce a enzyme structural protein that is required. Could be RNA, could be tRNA, could be anything that's required for the normal functioning, whereas the regulatory gene is to regulate, that's why it's called a regulatory gene, regulate the transcription of other genes. So that's the important thing of it's going to code for a transcription factor, or it's going to code, for example, for a repressor protein potentially. Okay, so let me just move on then from that one. And I want to bring in, I'm going to go genes just now. I want to just cover a bit about inheritance to see how we go on with that, because I do feel as well patterns of inheritance are coming through, and they'll either go down the route of a genetic diagram. I feel maybe that you've got to pull information out of that, or it's going to potentially be a application of the skills. How does conservation ex situ and in situ for module 4 link to conservation eco? So conservation ex situ and in situ, Elisa, if you're talking about that with ecosystems in module 6, you are looking at the fact that an ecosystem is about the community along, so the abiotic and biotic factors. If you're having to remove an organism, so if you're doing ex situ conservation, you're removing that organism out of their natural habitat, you could therefore affect the biodiversity within that ecosystem. If you're doing in situ conservation and you are helping to manage that area, you are therefore going to maintain, so specific treatments, techniques that are involved in maintaining the biodiversity. So that conservation, whether it's, if it's ex situ, removing it, you're only going to conserve that one organism. But if you're doing in situ, you're therefore not only protecting your endangered species, but potentially the other species within that ecosystem, whatever those species are, with that example. See, I talk too much, I end up not getting to actually finish my coffee before it goes too cold. Okay, let's keep going with this then. So again, this question's a really nice one. I love the fact that this exam board, I actually do like using different exam boards because they've got different ways of asking the content. But if you know your application skills, then you will be able to answer this type of question. So if we were looking at a question like this, so here we can see we have been given some information. So you should already know about sex-linked traits. So already they've told you in this question here that you have got a mutant allele which is found on the X chromosome. Now, remember, blood clotting is part of your immune response. So therefore, this could link to module 4, but also linking it to module 6, because we're talking about the inheritance of a mutant allele potentially. So they may look at, do you understand that if you're female, you have 2 X's, and if you are male, you have an X and a Y. And if we understand about hemophilia and the fact that you can have the capital dominant allele and then the recessive allele. You could be homozygous dominant. You can be, so therefore two capital B's. You can be heterozygous or you can be homozygous recessive. Whereas if you are male and it's found on the X chromosome, you only require one. So therefore you can either be dominant, so you've got that allele which codes for the dominant trait, or you've got the lowercase one. Now, this isn't what this question is actually asking about. This question is asking about, do you know how to pull information from a table, a novel idea that you've never come across before, and can you then link that to a topic area that you are meant to have studied and be aware of? And this one actually is looking at, do you understand genetic engineering? So this is where I love these types of questions, and this is why I want to show you this, because with these types of questions, what they're actually doing is they are trying to throw you off slightly by giving you some information. Your brain goes, "Ooh, I know about haemophilia. I know it's on the X chromosome. I memorized that." And then they give you completely different bits of information, a different question. So you start writing answer, which is completely irrelevant because it's not answering the question that's in front of you. And I can see how a lot of you may have done that in paper one's questions. So just be aware when you're going through this that you are taking the time to really read what the question is asking you and assessing you. So this first question is actually about genetic engineering. Do you know how genetic engineering actually works? Do you understand the principles? Do you know that if we're genetically engineering something, we are therefore taking DNA? So many people said nothing came up about DNA in paper one. Well, I feel like it's coming up in paper two. If we take the DNA from one organism and we are going to insert it into another organism, how are we going to do that? How exactly are we going to take the DNA from one and insert it into another? Do you know the principles? Do you know that you should be able to talk about whether we're talking about the DNA being cut, oh, I just wrote cute there, being cut specifically. If it's being cut, that DNA being cut, how are we cutting it? We're using endonucleases, aren't we? That's what we're using, right? No, you're not going to get the marking point for that. You should know the word restriction endonucleases. So again, some of you are saying to me that you're not getting enough marking points. And why is that? Do not try and limit your answers down to miss out some key words. Always answering restriction endonucleases. Well, that's only if we're cutting the gene out from the actual chromosome that it's found in. What if we have mRNA? What are we going to do with the fact that we may just have mRNA? Well, if we have mRNA, we then need to be talking about our reverse transcriptase. We also need to be talking about the fact that we have got our complementary DNA. What else do we need to talk about? We need to talk about the fact that we need our ligase to join the fragments together. What else do we need to talk about? Well, how can we join those fragments together? So ligase, this joining of fragments, is only because it's going to form phosphodiester bonds. So how did those two fragments come together in the first place? Those fragments came together because, and this is another thing, students will go, ooh, they have sticky ends. And they're like, yes, nailed that one, got the marking point. That's not the wording you want to use. They come together, the fragments, i.e. the cut desired gene, and our vector, which could be a plasmid, for example, come together because they have complementary base pairs. They form hydrogen bonds. So this is where I would say for some of you, you can trip yourself up by using generic terms that even your teachers may have used. Okay, so your teachers may have used that term, sticky ends, and it's not wrong in what they're saying, but in the exam, they do not want you to say sticky ends. They want you to use the scientific vocabulary. Complementary base pairs forms hydrogen bonds. The ligase is just going to join the fragments together by those phosphodiester bonds. Obviously, DNA being cut, is it restriction endonucleases or are we using reverse transcriptase? Now, I do feel like there was a lot of questions in your exam which were about enzymes. So I feel like they may ask, I mean, enzymes are involved in everything basically, but they may not go so heavy on enzymes in this next exam, but it'll be about the production potentially of the enzymes, but I don't know. As I say, I can only guess based on what I feel at the time, and I could be wrong. I could be wrong. I might be right. It'd be great if I'm right, because then I've helped you guys, but I might be wrong. But I want to go back to this question as well, because this is a, how do you pull information out of a table of results if you are given it? And this is a lovely table of results, because what they really want you to do is dissect the information that you've been given. So you have to take the time to read the information, i.e. 4.7 kilobase pairs long. So you've already been told that the virus is the vector in this case, not a plasmid. We've got a virus, which is a vector. And what we're going to do is in this vector, we are going to introduce potentially a gene, a desired gene, and we're doing it through the process of gene therapy. So we're inserting a gene to a vector. The vector is the virus, but the virus's genome is 4.7 kilobase pairs long. So then we need to decide, if we're going to do this, which of the treatments would we want to use? And this is where, by looking at the data in the table, what you'll be able to see is this first one, the gene F8, more people actually suffer with hemophilia A compared to hemophilia B. So if you were going to actually assess whether or not that would be the better treatment, they won't word it exactly that way. However, what they're looking for you to know is, well, this one is more common. Because it's more common, then actually maybe it's better to treat hemophilia A with that one, insert that gene, the F8 gene, rather than inserting the F9 gene. Or if we look at this, let's look at the base pairs. In this one, it's 1.6 kilobase pairs, whereas this one is 7.0. So therefore, this one will definitely fit in the virus genome. Because obviously 7.0 is a lot higher than the current genome, which is 4.7 kilobase pairs. So this case, what we're doing is we're having an argument, but we could give the argument for one, but we can also give an argument for the other one. And so therefore, the way OCR asks questions like this, it's a evaluate. Whereas in this question, they've said assess. It's the same thing, they just changed the wording. But what they've done is they've given you information for you to be able to pull it out. And then they're saying, okay, well, whether they should attempt to treat hemophilia with gene therapy at all. Should we treat it at all? Well, what are the risks? Have they told us anything about the risks? Well, they haven't told us anything about the risks. So that's something we need to consider before we say yes or no. Well, what are the risks one way and what are the risks the other way? So actually, it could be an argument for and it could be an argument against. What we also want to know is, well, is there a cure? So if there's no cure, then we would argue that gene therapy is a good form of treatment, at least attempting gene therapy to help someone who potentially has haemophilia. So it is important when you're going through these questions that you take the time to dissect that information so you can figure out what it is that they're actually asking you. And then let me just one thing that I thought was quite interesting with this question went on, I do want to just add it in here before I move on to the next topic area, is in gene therapy, the gene coding for the clotting factor needs to be introduced together with the promoter. Why does it need to be put in with the promoter? This is again checking that you know, let's try that again, that the promoter is where RNA polymerase binds. So to ensure that it is actually going to be transcribed, you want to have the promoter in there as well, is another way to look at that. And therefore allowing for the gene to actually be switched on. Oh, and this one as well, still looking at the same question, but taking it in a slightly different way. So this is, I really liked this question because of the fact that it does actually bring in your immune system in two different ways. Module 4, last year, they asked a lot about the different cells, but what they may do is bring that in again, but this time asking the questions in a way which is about applying your knowledge. So you're told in this question, some individuals who were taking part, oh, let me switch pens again. Taking part in the gene therapy trials are naturally exposed to a virus that is carrying the functional gene. Well, what happens when you're naturally exposed to a virus? When you're naturally exposed, you should know about active immunity. Once you have been exposed, that's going to result in you having memory cells. Those memory cells are going to recognize the antigen, and therefore you are going to have an immune response which will be able to fight against the virus which has the functional gene. So what's important for you to note here is that if they're asking a question like this, they're expecting you to pull that information forward so that you can say, how will this affect the success of gene therapy? Well, if we're talking about the success of gene therapy, we need to think about our immune system fighting against that virus. So therefore, we already have antibodies, we already have an immune response that's going to try and destroy and attack that vector. That's a problem. So we are going to therefore result in a decrease in the success rate of that gene therapy. So it's important to not just solely look at these questions as if they're only asking you about gene therapy and about looking at how the bigger picture applies here. Okay, so I'm going to move on now. And I want to talk about speciation. I'll lift one in there. And looking at the formation of a new species. So obviously, if you guys have watched my videos, you'll know that there's lots of stuff I have on different isolating mechanisms. If it's allopatric, different environments, geographical isolation. You should know about sympatric speciation, reproductive isolation. You should know about what that means in the fact that if we have different numbers of chromosomes, that's going to have an effect on the ability of the plants from being able to reproduce. So therefore they become reproductively isolated. So just to keep you in the loop, reminding yourselves, module 2 is about mitosis and meiosis. So this is where they could bring in mitosis and meiosis and assess your understanding on that if you're looking at how the reproductive isolation comes into play. Because if you have an organism which is having a different number of chromosomes, if we're talking about animals, the animals are going to be less likely to develop, and therefore we don't tend to get as many hybrids in animals than we do in plants. And how do we know that? Well, this is where we can look at classification in the fact that there are so many different varieties that have evolved from the same families. So again, that's where they could bring in classification and evolution. So what I wanted to show you, and I don't know why I've got a strong feeling about this, but remember that speciation, this formation of a new species through that process of evolution, you're expecting some form of isolation, some form of random mutations that occur through spontaneous mutations due to DNA replication, or you have some mutagens which can accelerate that. So they could look at module 6 mutations, but in the form of the topic area about speciation. So it is a bigger picture here. But you've got this random mutation. Is that random mutation going to be one which is a silent one? Is it a missense one? Is it a nonsense one? What are the consequences? Is there a frame shift happening? What is happening? But whatever you have, that random mutation, you're going to have different environments, different selective pressures, different characteristics are going to go for and against. So we're going to see a change in allele frequency. Hardy-Weinberg equation coming in potentially as a question this year. I feel like they love a Hardy-Weinberg equation. If there is no longer an ability for them to interbreed, they are now reproductively isolated. They are now new species. So this is the question I want to look at, which is a really interesting one. I know they did a lot on PCR last year, and they did a lot on the bioinformatics and computational biology. However, I do feel like they could still bring it back in again. So just be aware, they may bring it in, and what they're actually doing is they're assessing your module 2 knowledge on, do you know the structure of DNA? So do you know the structure of DNA? Do you know how these different temperatures are going to help in that process of allowing that change so that we can amplify the DNA? So what's the difference between bioinformatics and computational biology? So bioinformatics is about building the databases to store the biological data that you have got through DNA sequencing. So you're talking about storage of the data for comparison. The computational biology is where what they do is they take that information and they then make simulations, they do models of it so that they can work out and predict using statistics, predict the likelihood of that, for example, gene or that combination, that sequence of amino acids coding for the folding of the protein that can then have an effect on, let's say, the ability of your brain cells to retain information. Just giving random examples there. So the informatics part, bioinformatics part of it is the information, how it can be stored and how it can be held so that it can then be compared. Yeah, it did come up as a 6 marker last year. I think it will come up potentially as a question, but if I was to put my gut on it, I'd say they're going to ask it in a multiple choice question and not as a long question, or it could be a 1-2 marker, but nothing more than that, I don't think, because it was such a big 6 marker. But going back to this question, so do make sure you know this question is looking at, do you know DNA replication? So if they're asking a question like this, they're actually checking your module 2 knowledge, and they're checking, do you know why the Taq polymerase actually has to go through those cycles. So obviously they've given you all the information you needed for PCRs. They're not interested in that. They're interested in, do you know about the fact that you have hydrogen bonds, and those hydrogen bonds between your nitrogenous bases are going to need to be broken. So we need to break those hydrogen bonds. I would say, Sahib, they can ask it about anything, to be honest. It just depends. And that's where a lot of students fall down because they're like, oh, well, they asked it last year. They're never going to ask it again. They will ask it just in a different way. And so for 23s and 24s paper, students were so sure that excretion wasn't coming up because it came up in 23s. They were completely thrown, but the question that they asked in 24's paper was actually about the linking it to animal responses and hormonal system. So they can ask the question whichever way they want because you're meant to have learned the whole course. So that's why I don't like doing predictions of what I think is coming up because I could tell you one part of it, but they could go off on a tangent and ask it in a different way. So it is that trying to figure it all out is hard. And so make sure you are revising all the stuff that you hate, because if that comes up, then at least you feel a little bit stronger. And if you're just revising the stuff you like, yes, you're consolidating it and you'll remember it, but they may ask it in a weird way that you can't apply it. And so that will throw you off as well. Anyway, back to this one. 95 degrees because we need to break the hydrogen bonds. 50 degrees C because we know that, or you should know that DNA polymerase cannot work on its own. Yes, it builds the phosphodiester bonds, but what you need to know is it needs a primer to begin with. And so by dropping it down to 50 degrees C, you're allowing for this primer to form complementary base pairs. And therefore, the DNA polymerase can then build from that primer at the 72 degrees C. And so the DNA polymerase can build the new strands, and it does it over and over and over again. And so that's why I would say that. I feel like in skills, those topics are very poorly taught because they are somewhat vague. Well, Sahib, I would agree with you on that one. And I have to say that's why I built my YouTube channel, because a lot of my private clients that I tutor come from lots of different skills across the UK. And therefore, I realized that there were moments where some students were being disadvantaged, and that's not to slate their teachers at all. It's because within the teaching profession, we have to not only teach this subject area, i.e. A-level biology, but we have to teach so many other things, and there are other processes we have to do. The space in our brain to be able to retain or learn all the information is actually highly reduced. And so I took 6 months out to build the YouTube channel for you guys because I realized there was nothing that I could direct my students to, or there was nothing for me. When I started this 2 years ago again, I hadn't taught this and they changed the specification. I had to go back and relearn it. And so I did the same thing, that I had to relearn it and I couldn't find good resources. So it's really hard because for some of us, we have really strong content knowledge in specific areas and we're really hot on that. But you ask us something else that we haven't taught in a couple of years or over a summer, and it's literally like it's gone out our brains. So it's a difficult thing, but that is why I built the YouTube channel, so that it could be there to be used, and hopefully people find that it gives them the information they require, but it's still a massive learning curve. Like my partner works in bioinformatics. He's a computer science engineer, and I've learned so much through the last couple of jobs he's been working at because they've all been based around working alongside bioinformatics and statisticians. So I've learned so much more about it to then try and bring that into my teaching. So it is difficult, but it's in the spec, so therefore they can assess it and they will assess it in whichever form they decide. I could feel it was coming more and more through the last couple of years, hence why there was such a big question on it this year, I feel. Anyway, let's move on, because what I actually want to really focus on is this question here. This is the sort of thing that I feel where they could take you with the question if they're going to ask a question, because it's again application based, and this one is looking at the fact that you've been shown a diagram. Now they're telling you you're in New Zealand. Okay, so we're in New Zealand islands, and we can see that there are different islands. We've got the North Island, we've got the South Island, and we've also got the Stewart Island. So there are 3 different islands that we're looking at here. So straight away in my head, I'm thinking to myself, ooh, different islands. We have got geographical isolation. I haven't looked at the question yet. I've just thought to myself, what is actually being shown already? So the fact that they are showing me geographical isolation, I have some form of isolating mechanism which has occurred because something's in the way, whether that be a mountain, whether that be a river, whether that be a sea. Geographically, these species have been isolated. So that's the first thing that I am keeping in my mind. So then what I need to do is I then need to look at this and say, right, what else have they told me? They love giving you the information to pull out from there, but it's about your comprehension. Can you read and pull the information out from what they've told you? So what have they told us? Let's just have a look. So they've told us that we have got in the North Island. Oh, come on, pens. So in the North Islands, there's mainly lowland habitat. So we have a specific type of habitat, which is lowland. Then they've told us that in the South Island, and also, and you see how I'm highlighting here right now, the Stewart Island, these ones have got mountains and alpine habitats. So they are already showing that there are different habitats. We have different habitats involved here. So therefore, that is something we need to note. That could be a way in which we are geographically isolating them, but we need to be sure. So let's just see what else they've told us. DNA sequence data. Well, where did that DNA sequence data come from? DNA sequence data had to come from DNA samples. That DNA sample would then have to be amplified using the PCR. And then what else are we going to do? We're going to sequence it because we're going to be using our terminator bases. Alongside the terminator bases, we're also going to be having fluorescent dyes. Dyes. So all I'm doing is I'm just kind of triggering in my brain, what do I already know in case they ask me questions on that. I don't know where they're going to go with this question. All I can do is try my best to prime my brain to think about the ways in which I could answer this. Okay, let's keep going then. We've got 3 species. Ooh, interesting. 3 species. So they've all got the same, remember what we have here. There's my genus, there's my species. So they're all within the same genus, but we have different species that are going to be compared. So again, this is classification potentially coming into this question. I don't know. It could be evolution because we're looking at the fact that their DNA is genetically similar or genetically different. Did they have a common ancestor? So this is where, if we're looking at OCR's way in which they'll assess this, they'll give you a bit of information and be wanting you to pull forward that knowledge that you already have. So let's just finish doing this. So what have we got? In the alpine species, individuals on, what color did I use? Green. Oh. The South Island showed genetic differences from individuals on Stewart Island. Well, that's obvious because they've been geographically isolated. So the fact that they've been geographically isolated because one's on the green island and one's on the pink island, they're geographically isolated. Okay, so now they're potentially in different environments and therefore have different selective pressures on them. So in my head already, I'm starting to think about my keywords. What do I know? Well, I know that I've got my isolation that's occurred. And then I need to now bring in, well, once they've been isolated, there has to be some form of selective pressure. Some form of selective pressure, which has resulted in there being those that survive and those that die out. But hold on, where did that come from? Oh yeah, mutations. So mutations will have occurred to give us variation. Because of those selective pressure, some are going to thrive and survive, reproduce, others are going to die out, and therefore we're going to have the allele frequency change. And that's going to result in that changing occurring where we end up with different species. So we start off with common ancestors, and then we are diverging away because of these mutations and these selective pressures that are going on. Well, we know that because they're in different islands, they've been geographically isolated, they have some form of selective pressure. So that's the first thing we need to know on that first one. So we've done this one, right? In the, where are we now? In the alpine species, so we've got a new species there, individuals from different mountains, but this time we're on the South Island and they're showing genetic differences. So this time we've not got geographical isolation because of seas or oceans. We've got geographical isolation because they're on different mountains. They have been isolated because of the mountains that are present, and therefore, again, isolation, selective pressure, there's been some form of mutations, spontaneous mutations, causing variation, allele frequency changes, because some that are better adapted will survive. They will therefore be able to reproduce because they can reproduce, they can pass on their alleles. The population's frequency of these alleles will change in potentially the direction, and I'll come back to that in a minute, the direction of whatever it is that is best for the environment. But now let's look at this last one. So that's us done our second one as well. In the lowland species, right, okay, so we're in the lowland species here. Individuals from different areas on the North Island, these guys show genetic similarity. So therefore, what we're saying is their DNA sequences are similar, therefore less mutations. Therefore, we can make the prediction there's been no real form of isolation that's occurred and no form of isolation. Therefore, there was no need for them to actually go their separate ways. So interbreeding is still possible. Interbreeding is still possible because they have DNA sequences which are similar, and therefore we can say there's less mutations have occurred. So they've given us all the information we needed in that stem of the question. But a lot of the time, students, even myself, the minute I see a question like this, I go, and I've had to train myself to not panic, to slow, and I am a slow processor. I have to slowly read the information and really dissect it as I'm going so that I can then help myself to get that information forward. So do make sure when you are doing this that you are taking the time to think about what might they ask? And the other thing they may ask, they may bring into this question, is they may ask you, as they have done in this exam, to think about other factors, 2 factors other than natural selection that could drive that genetic change. Because remember, it's not just about natural selection. Discuss is like them saying evaluate. Sahib, if they said a question like discuss reasons for the results, discuss what you think you can get from this. As I say, this is not a OCR A question. So therefore, if they're saying discuss, basically arguments for or against, treat it like an evaluate question. Discuss what you can see from the information. Well, I can see this and I can see that. I can see this and I can see that. So whenever they are asking that discuss, it's like treating it like an evaluation. So going back to his last question, then 2 other factors that could drive genetic changes. Well, this is where you could bring in your genetic drift. Didn't even work there. Oh, hold on. Try that again. Genetic drift. So you could say genetic drift, you could say the founder effect could have a role to play. Now, I am Scottish. I am originally, actually, I was born in England, but then my whole family are from Scotland. I lived in Scotland for most of my life. However, up until the age of 6, I lived in Germany. So the fact that I was in Germany, the fact that I've moved from Scotland to England, I've been here for over 20 years. What does that have to play with this? Well, you should know that organisms can migrate. So migration could be another factor other than natural selection that could drive genetic changes in populations. Mutations, absolutely, you would get mutations as an answer for that one. So mutations is obviously one that we could add. Oh, it's done it again. Mutations is another one where I could answer that one. When you say time though, Sahi, what do you mean? How would time have an effect to play? So Amna, with predation, you are talking about the fact that they're going to be preyed upon. That would therefore decrease the numbers of those who are less able to survive. So that would therefore link back to the natural selection. A couple of years ago, asked this question, and the mark scheme just said time. Say 2 factors of the natural that could drive genetic changes. I think if they were going to give it, time alone wouldn't be enough. Basically, what they're trying to say is they're like, oh, okay, they're kind of right. We'll give them an answer. But yeah, I would say time without any other reason behind it wouldn't be enough, I don't think. I think the examiners this year would be like, that's not enough. So you'd need to be saying about 2 factors that could affect the drive in genetic changes. Yes, time is true, but good selection. It's me putting 2 words together. Sexual selection, absolutely. If we're talking about selective breeding, that could have an effect. Absolutely. That would be the other way in which you could answer that question to give that to you. Right, how long have I got? 12:42. I don't even know how long I've been going for. Oh, an hour. I've been doing an hour. Okay. What do I want to? I want to just jump in. Let me just put in. Let me bring in some stuff on. So they're the kind of questions that I thought were really good questions that I want to go through, but some content I do want to cover. I have a strong feeling, and again, I could be completely wrong on this. I have a strong feeling the aseptic technique is going to come in, and also fermenters potentially. So if they do come in, make sure you understand that you're talking about culturing bacteria is not just about culturing the bacteria. They could ask it about the microorganisms and linking it to the growth that they want, depending on where they want to go. So although we're talking about aseptic technique here, and I'm going to talk about it with regards to prokaryotes, they could ask it linking it to yeast, okay? They could link it to some other microorganism. So just be aware of that. But what's important? Why do we study bacteria? Obviously, large numbers, we can grow them in culture, whether that culture is a broth, as in a liquid medium, or whether it's a solid medium, such as agar, then it doesn't matter. We're growing the microorganism, we're culturing them. So make sure that you are aware that they could give it to you, either talking about it as a broth or as a medium. That if they go down the route of the prokaryotes, it's binary fission. If they're talking about eukaryotes and they're going down the route of yeast, you know it's a fungi. Yeast is a fungi classification, so you also know it's eukaryotic. If it's eukaryotic, you know that it would be mitosis. So do make sure that you're checking whether they're talking about prokaryotes or eukaryotes in the question. And when they say here that with the prokaryotes and they've got enough nutrients, temperature, pH, oxygen, what they're looking for is, do you know what those nutrients are? So what do they need? Do you know that it's carbohydrates, lipids, and proteins? Do you know where those sources of nutrients can come from? i.e., is it a monosaccharide? Is it a disaccharide? Is it a polysaccharide? Which are the best nutrients for them to actually provide for the microorganisms to grow? So this is where they could link in biological molecules under the concept of fermenters, okay? Why is temperature important? Temperature, straight away, you say thermal energy, kinetic energy, more successful collisions. Therefore, you'd expect a faster rate, and in this case, a faster growth. pH, why is pH important? Because we know that proteins can be, especially if they've got a tertiary structure and they're globular, that they are going to be pH sensitive, and therefore they could denature above or below. Now, it's an interesting one because they didn't do, I'm just reminding myself when I did paper one this morning, I didn't see any question which was looking at denaturation of enzymes to do with pH or temperature. They did have inhibitors in there. There was inhibitors, but there was nothing. So I'm just scanning through my brain of all the stuff I went through, of where my thoughts are going. They didn't have anything on pH and having an effect, or temperature and having an effect, which would therefore make me be a little bit more confident that they could bring in the fermenter and the fact that the temperature needs to be controlled. But I'll come back to that. And obviously oxygen required because you want aerobic respiration. But one thing I would say to you guys is that as I'm doing this, as I'm thinking this through, these are me just going on my gut judgment based on the content and based on what I can see and what I believe. As I've said to you, this doesn't mean it's definitely coming up. So don't think that what I'm telling you is gospel, that that's what they'll ask. I don't know. I basically have had everybody email me with all the information of what they remember coming up in paper one and the comments on my videos, but I only got to see the paper this morning. So I didn't get any access to the paper whatsoever until this morning. Now I've seen it. Now I've seen the way they've asked the questions. I can then think about it, but I don't know. I'm just having a guess. So please, I'm just putting that out there, that this is just me telling you where I think they could go based on my own research. But it doesn't mean it'll definitely come up. So please don't think that I am telling you what I know is coming up, because I don't. I didn't write the paper. I mean, I write many papers, but not for OCR and not for the exam boards anymore. But through my role of being head of biology, it was something I did on a regular basis. So I've used that knowledge to help in this case. So I just want to make it very clear, I do not know what is coming up. Anyway, back to this. Asepsis. sterile environment, techniques, inoculation, what we're going to do. Obviously, we're going to flame it. We're going to make it red hot. We don't want to kill. Important here, we want to kill any bacteria we don't want, but we don't want to kill the bacteria we do want. So we have to be very careful that we don't take a red hot inoculating loop or spreader and stick it straight into our growth medium that we want to utilize. So this could be a question where they look at the, how could you improve? Students did a practical where they were using aseptic technique and they were spreading it out and they were trying to grow these bacterial colonies like you did for your PAG, but they didn't have any growth. So if they got if you got a question that said they didn't have any growth, how could they improve this practical? It could be that they put a red hot inoculating loop or spreader into their broth medium, and therefore they have actually killed the bacteria they want to use. So that's a way in which they could check your practical skills and how you apply that. Okay, back to what was I talking about? Flame the tube, flame the inoculating spreader. Also, you've got this convection current that's going to occur. If you've got a Bunsen going on, what you're going to have is, as that Bunsen is heating up the air particles around it, they're going to spread out more because they've got more thermal energy, means more kinetic energy, more movement of those particles. They spread out, they become less dense, they rise up. As they rise up, what's going to happen is that's going to pull air, colder air, in to replace that air that's just gone up. So you end up with this convection current. So if you leave your Bunsen going for a period of time, then what's going to happen is you're going to create a little sterile environment around you. And that therefore is going to ensure that you are making sure that there is no other contaminants around you, other bacteria which could be in, or even fungal spores. Remember, that's the other thing to remember, that how do they reproduce? If you've got fungal spores and those fungal spores are transported in the air through the wind, through air particles, they could then fall and contaminate your plate. So again, this is how they could kind of bring that back round again. Make sure, now they did do logarithmic scales. I saw that graph. That was a really interesting one. They did do logarithmic scales in your paper 1 exam. That doesn't mean that it's not coming up again. It was about you pulling information from that. That question actually took me a while. But what I would say, that's because I hate maths and I was like going back and forth with my guessing on it. And I want to double check every single part of the graph. But make sure you know your lag phase, your log phase, your stationary phase, your death phase. Make sure you know those phases. But what I would say, where I feel they did a lot on limiting factors in paper 1, doesn't mean they're not going to bring limiting factors back in, but limiting factors such as nutrient available, oxygen, temperature, buildup of waste, change in pH, they're important when you're looking at fermenters. So obviously with biotechnology, we're going to be using organisms, fungi, yeast, and bacteria, or we might just be looking at enzymes. So if they were going to ask a question about immobilized enzymes, which actually they mentioned immobilization when they looked at that photosynthesis question. So I don't know, my gut's kind of changed on that one, but and because they've done a lot of enzymes on that as well. So I'm not sure if they're going to go down the route of enzymes, but I feel like they could go yeast, they could go bacteria, it just depends where they go. The microorganisms, short life cycle, it's cheap because if they are going to reproduce really quickly, that's therefore going to result in us being able to get the materials that we want at a much faster rate. So therefore less energy needed to heat something up, for example, less energy, less materials needed because they don't require lots of things. They can be genetically manipulated. I can see where genetic engineering is going to come in, potentially. Sorry, guys, I'm not even answering any questions here. Let me go and have a quick check. Do we have any prediction? Could you say budding for? Yes. Alyssa, yes, you could say budding for yeast. Absolutely. Can you go over question 20? It's on fermenters. Yes, Sahib, give me 1 second. Let me just finish on this one, then I'll go to that one. Do they expect us to label a fermenter? They could. Random, they could actually expect you to label a fermenter, or they could expect you to describe why the different parts of the fermenter are needed. Like, how could you tell if it was A or B? Because basically that's what I actually did was I went and did the calculations, which is why it took me so long. But it's done, it's dusted, so you don't need to worry about it. Yeah, you did have to work out a gradient on that one. That would be my thing, which is what I was doing. Right, let's just, oh, crumbs, didn't mean to do that. Let's go back here. Right, moving on. So they could ask you to do the formula of number of individual organisms. Again, I've got the video on that, so do watch it. And it tells you how to go through this. So this where they could look at giving you a graph. It doesn't look like this. The graph that if they're going to give you a graph, and I think this is the one that you're talking about, Sahib, and I will bring the paper up. But if they're giving you information about, for example, glucose, or it could be maltose, it could be starch, it could be lactose, it could be fructose, whatever they give you as the nutrient source, know that that nutrient either may have to be hydrolyzed down or that nutrient will be utilized straight away. So that could be um, could be a way in which they ask the question to check again your biological molecules knowledge. The secondary metabolites and the primary metabolites is an important one. If they are talking about primary metabolites, these are things that are needed for essential functioning. So when you do glycolysis, or when a microorganism does glycolysis, when it takes the glucose down into the pyruvate, that pyruvate is a primary metabolite because it is an essential part of normal functioning. ATP, essential part, coenzymes, so wherever they go with that, if it is produced during the active growth, i.e. the log phase, then they are primary metabolites. Secondary metabolites are not essential for normal growth, but they are used by the cell and they are formed during the stationary phase. So this is important for you to note. Where might you need to stop, for example, a batch fermenter, so that you can then collect and process those secondary metabolites. So if you know stationary phase for secondary metabolites, and you know your log phase is your primary metabolites, then when you look at, let me just skip through here, these are all, by the way, on my channel specifically if you want to look at it. But if we're talking about batch fermentation, we need to talk about the culturing. Not only do we need to talk about the culturing, we also need to talk about the fixed volume going in. So we have done our sterile aseptic techniques to get our microorganisms at a fixed volume. Then we're going to put it into our bioreactor, which is also going to have to be sterilized. We're going to stop it during the stationary phase. What are metabolites? Metabolites are the end products of chemical reactions. So a metabolite, so for example, pyruvate is produced at the end of glycolysis. Pyruvate is classed as a metabolite. Pyruvate then goes on through the link reaction to produce our acetyl coenzyme A. Acetyl coenzyme A is our metabolite. So a metabolite is a chemical that's produced through a chemical reaction. Does that help? Right, I'm gonna move you away. And then going back to this stationary phase, so we can get that desired product, whether that's an antibiotic or whether it's an enzyme. Now, remember that I did mention, let me just find where this slide is. Where are you? There we go. Yeah. I did mention the fact that genetic modification, I feel like they could bring in genetic modification here, in the fact that if we are taking a desired um, product that we want. We're taking the gene for it, we're inserting it into the vector, the plasmid, and then we are transforming the bacterial cell. We are therefore going to um, help to give the help to give, what am I even saying now? We're going to insert the gene. We're inserting the gene to the vector, which is the plasmid. That is then taken into the bacterial cell through electroporation. After the bacterial cell has taken that in, it will then go through binary fission, which means that we then have multiples that have the inserted gene. We are then going to stop it at the stationary phase when it's making our desired product that we want. It could be insulin, it could be an enzyme, it just depends whatever the question asks. But we genetically modified the organism to produce what we wanted. Okay, so that's where they could go down that route. Obviously, stopped before the death phase, and the product harvest and the system sterilized. The continuous fermenter. Now, this is an important one. Asepsis, bioreactor sealed, aseptic unit. We're going to continually add. Now, this is an interesting one because they love throwing this in there. Why are we giving them ammonia? What's the point in ammonia? Why not just glucose? Or what's the point in glucose? So if they give you a question that talks about ammonia, you therefore know that this is going to be needed to make amino acids and therefore make your proteins. So because they are giving you a little bit of information, that helps you. Absolutely. Provides the nitrogenous compound. Be careful, Sahib. It's the nitrogenous compound, and therefore the nitrogen that can be used, so you can add in the amine group, amination. And also, absolutely, nucleic acids, because remember, DNA replication. DNA replication requires nucleic acids. And again, that's where some students miss that bit out because they do go amino acids and proteins. Proteins for growth. It's as if you knew where I was going to go with that. But the DNA replication and nucleic acid side of things is important. And it's why I've said nitrogenous compound. The minute you see nitrogenous compound, you should be thinking about all the things that have nitrogen containing compounds. So a nitrogenous compound could be RNA, could be DNA, could be amino acids, could be proteins. So don't just get caught up in the amino acids and proteins. Try and use that to help you. Glucose, why is glucose needed? Well, we know that that's going to be for respiration. And that's therefore going to be producing the ATP molecules. And those ATP molecules are going to be used through the process of, if we need to do protein synthesis, if we're making proteins, we need protein synthesis. Therefore, this is an energy demanding process. Because it's an energy demanding process, they require ATP molecules. Well, if they're doing respiration, if they're doing metabolic reactions, we know that it's going to heat up. If it's going to heat up inside the bioreactor, that is therefore potentially going to get too high and could only after we have gone past the optimum level, only once we get past that optimum level, will we therefore potentially have denatured the enzymes, and therefore we need a cooling system. So it's important you note that we're going to add in the nutrients for that exponential growth. We're at the log phase. We're going to ensure that there's mixing inside. Now I haven't drawn it in my in the picture that I've got here, but what I should really have is what they'll have in the exam is it's kind of like a paddle. And that's going to evenly distribute not only the heat, but also the molecules that are being added. So the glucose, the ammonia, oh, air. Why was there air? Oh, yes, oxygen. Why is there carbon dioxide? Oh, yes, that's a waste product. So it's important that we know that it's continual fermentation, continuous, because we have a culture broth being constantly removed whilst we're also inputting the nutrients that are required. So we're continually adding stuff in, we're continuously removing not only the stuff we want, but also some cells, some waste products, etc. And therefore, we're going to need to do downstream processing. We need to take out the useful parts, we need to separate them. Removal of waste. If we don't remove carbon dioxide, we're going to have affect our pH, because we know water and carbon dioxide will react to form carbonic acid that will therefore dissociate to form hydrogen and hydrogen carbonate ions. Interesting, hydrogen carbonate ions came up a little bit in paper 1. But what we have to remember is this is a reversible reaction. So because it's a reversible reaction, it means it can go back. So we've got the hydrogen ions, which are going to affect the acidity, but the fact it's reversible, we can remove the carbon dioxide and therefore ensure that we're not affecting the pH, because remember, proteins, if they are globular proteins, if they have a tertiary structure which is sensitive to pH. So we want to ensure that if it's the proteins we're looking at, that we're making that pH not change too much. So what they may also ask you is about how could you monitor this? And this is where you'd say you'd have a pH probe. And then obviously the temperature, you've got your rate of reaction. Rate of reaction is going to increase if the temperature is increasing, but if we go too high, we're therefore going to affect that rate of reaction. So they can go either way they want on that one. So because of the property of water, because water has a high specific heat capacity, that therefore is going to ensure that we are able to maintain that temperature, that it doesn't get too high. Somebody asked me about bioremediation. So other things that you could be asked about. So examples, you do not need to recall these. As I said before, they've removed bioremediation, that you don't need to recall them anymore, but they could still ask questions about it. So if they do, breakdown of pollutants and contaminants, fungus grown in large fermenters with glucose syrups, and then added to albumin. Sahib, you did ask me about a question. What was it? 21 CI from the 2022 paper 2. Let me just have a quick look. And then I'm probably, guys, going to finish there for now because I have been going for quite a while. I will jump back on here at another point when I have a moment. Right. Paper 2, 20. Paper 2, 2022. We're looking at 21 CI, right? Exactly. That is exactly the question I was actually referring to. 1 second. So if we go So this is exactly why I asked these questions, because this is straight from, you're absolutely right. There you go, right, hold on. I'm going, nope, I don't want to do that one. Hold on, how about this? There we go. So that was the question that they actually asked in that exam, using the diagram to name the type of fermenter being used for the mycoprotein production. And it was exactly that picture that they used. Hold on, where's it gone? This one was the picture they were referring to. And they did ask specifically about the name and justification. So use the diagram to name the type of fermentation process used for mycoprotein production, and then suggest why the cooling. And then they also asked about the air inlet. But absolutely. And that's exactly why all my resources that I have built have been based around the exam questions from OCR, so that you can actually apply your knowledge to that question. So which one? Justify your action. The fact that they're putting in the nutrients, they're removing the products, the waste products, as well as the product collected down here, would tell you that that's the continuous fermentation. And then suggest and explain why a cooling system is necessary. Chemical reactions results in thermal energy being released, and that will then heat things up, which could affect the enzymes. And then the air inlet provides the fungus with oxygen for respiration. Ammonia. Suggest and explain why the fungus is provided with ammonia, and that is, as we said, was to do with the proteins, nitrogenous compounds, etc. Right, guys, thank you for watching and staying with me. I know it's a long one. I'm trying to get as much out there for you. But I do feel like if I were to make any gut judgments right now, I feel like gene regulation, absolutely. I feel like patterns of inheritance, natural selection, and speciation potentially, but definitely Hardy-Weinberg, I feel, is coming in there. I feel like genetic engineering. I feel like, what else have I said? On my list. I've got lots of different lists here. What else have I got that I've not mentioned? Batch continuous fermentation, lac operon transcription factors, natural selection, speciation. Oh, plant diseases, I feel, may come up because they did animal diseases last time. I feel like they may go down the route of plant diseases, but I'm not sure on that one. They didn't do anything about dissections, although they did show you a dissection picture. But it was like just a diagram to say it was dissected. So I feel like you've got a biological drawing coming. I do feel like nitrogen cycle and not cardiac cycle, carbon cycle could potentially come up. Percentage efficiency of energy efficiency. What would be different about the diagram if it was a batch one? If it were batch, you would have fixed volume. So you wouldn't have this part, you wouldn't have this part, and you wouldn't have that part. And you wouldn't actually need that either. So you would literally just have a bioreactor, and it would still have your paddle in between, and it would still be sterile, but it wouldn't have an inlet because you're using a fixed volume. So interestingly, let me just bring back down a second this one, because I skipped past it. So sourdough starter is a really interesting one because it's like a very basic batch fermentation. So basically what you're going to do is you're going to put yeast, you're going to put flour, and you're going to put water into a container. So this was, my partner had this hobby of making sourdough, and every so often we go back. But over COVID, when we were all at home, he learned how to make sourdough bread, which was amazing. But one of the things I thought was really fascinating is because this is actually looking at with sourdough, you could look at the fact that it's anaerobic respiration. If it's in a container, after a while, once the oxygen level has decreased because it was aerobically respiring, but after a certain time, if there's no more oxygen there, because you are batching it, you're doing it as a sealed container, it's going to switch from aerobic respiration to anaerobic respiration. So this is going to result in alcoholic fermentation. So they could bring in whether it was aerobically or anaerobically respiring based on was it using oxygen? Did it produce ethanol? So do have a look out for that as well. They did that 1 year, but it was a paper 3 question where they gave a graph of the carbon dioxide, the oxygen, the glucose, the ethanol, and they talked about the fact that the ethanol could be converted into acetyl-coenzyme so that it can then be go into the Krebs cycle. So do know they can look at your respiration. In fact, respiration can come through in any place they want, but in this one, yeast, flour, water in a container which is sealed, that is there for batch fermentation. So we've got batch fermentation occurring, and because of that, what we needed to do, or what he needed to do, is every so often he would have to open this, and when he opened it, he would therefore allow air to get into it. If the air is getting into it, it's now no longer anaerobically respiring, it's back to aerobically respiring, because if you didn't open it and maintain it and keep an eye on it, what would happen is the ethanol concentration would get too high. That would then result in the death phase. So if you're going to use batch fermentation, you want to ensure that you are not getting it to the point of the death phase. You want to keep it at a level where it's making the product that you want, the desired product, which would be the secondary metabolite, if that's what they want to ask the question with. And then the other thing, just to note on this one, it doesn't just have yeast in it, it also has bacteria. And actually, the bacteria, lactic acid bacteria, they are the ones which are going to produce lactic acid because they must have enzymes. Therefore, the enzymes come from your genes. The bacteria, therefore, must have the genes for making the enzymes. And so that's what adds to your flavor and texture to your sourdough starter. So again, it just depends which way they want to go in the questions. But guys, I'm going to stop there because I do need to have some lunch. And also, you guys need to take some time away, have some food, a break, and obviously make sure you're studying all the different things, not just biology. 'Cause if you are doing chemistry, maths, etc., you've still got exams coming up. So good luck. I will try and jump on as much as I can. Anita, I'm just finishing now, but next session, if you are in, then please do remind me and I will jump on. But I have done videos on my channel that have zero dilution in them. Go to biotechnology playlist and look at culturing microbes. I believe it's in there. But thanks,